Dear students, welcome to another video. This is on chapter 8. The Lac Operon is a classic example of gene regulation used by almost every textbook in biology. The contents discussed in this video are pretty taxing and will represent the pinnacle of your understanding in this course. It's important to understand that this is one example of the way in which bacteria can regulate their genes. There's another video on the tryptophan operon that will be released alongside this one, and that shows an alternative mechanism used for other genes. Please keep in mind that scientists have discovered many more mechanisms which are unique in the way that they manage gene expression. These three learning objectives are tied to this video. The first one will allow you to demonstrate one simple and elegant way that bacteria determines when to activate genes. So this is the baseline example for the Lac Operon. The second learning objective requires students to be able to deduce what normally happens at the Lac Operon regulatory DNA sequence. And finally, just to make sure that students have a good comprehension of the Lac Operon, we are going to move on and look at some mutations which can affect how the expression of the Lac Operon genes takes place. In order to relate to the contents of this video, it's important that students are familiar with the following terms. So gene regulation is basically gene control, how genes are turned on and off and by how much. As we've determined in the past, each gene has a promoter. Promoters are sequences to which RNA polymerase must bind in order to transcribe that piece of DNA. Operator sequences can be considered as blocks to the passage of the RNA polymerase. So on the DNA, there may be a sequence to which other proteins can bind and block the progression of the RNA polymerase. Finally, at the regulatory site, there may be sequences on the DNA close to the promoter that are known as activators. These sequences bind other proteins which aid, that is, help the RNA polymerase find the promoter and then start transcribing. The next three important components are proteins themselves. So these proteins interact with each other or with the DNA. So RNA polymerase, as we know, is a large protein which has the ability to make messenger RNA from one strand of DNA. The repressor protein will bind to the operator and prevent the RNA polymerase from either binding to the DNA in the first place or moving along the DNA to transcribe the genes. So this is a negative effect protein. And then finally, we have the activator protein. An activator protein is a positive effect protein. It binds to the DNA and encourages the recruitment of RNA polymerase to the site of the promoter. Lastly, we have some important molecules which make some important decisions as how and when to activate these genes. So since we're talking about the Lac Operon, it's quite appropriate that we talk about lactose itself, the sugar. Incidentally, allolactose, a slightly altered form of lactose, is what's actually being detected by the cell. But as far as we're concerned, we can use the word lactose. Likewise, when it comes to glucose, glucose has an impact on the Lac Operon, but it's not glucose directly that impacts the Lac Operon. It's one of its side players called cyclic AMP. And we'll see how that works shortly. It's quite fitting to introduce two very difficult concepts for students to grasp at this point. The term transfactor can easily be applied to any material that's wandering around the cell using Brownian motion. Proteins, as we know, are made by ribosomes, and in the case of cytoplasmic proteins, once these proteins are released, they can migrate around the cell unless they are tethered to the cytoskeleton. So these migratory proteins are referred to as transfactors, i.e. they don't have a fixed location within the cell. On the contrary, cis elements are fixed components within a cell. So if you were listening carefully, we use the word transfactors versus cis elements. And factors are normally proteins, and elements are normally regions on the chromosome as part of a gene. An example of this would be a promoter element tied to a particular gene. So the important thing is, unlike trans factors, these components are unable to float about the cell and thus have a fixed location. Now that we've finished addressing some of the foundational components in order to understand gene regulation, let's turn our focus to the Lac Operon itself. Research has indicated when cells are presented with alternative supplies of food, normally one type of food is digested before another. This is particularly true for E. coli. E. coli is a common resident of the human digestive system, and as we eat different foods, the E. coli is subject to differing environments containing that food. This research indicates that in all cases, glucose is the preferred source of sugar that the bacteria will break down for energy reasons. The reason glucose is chosen amongst all the other carbohydrates for energy purposes is because it requires the least amount of energy to break down the glucose and extract that energy from those covalent bonds. Thus, when glucose is available, the genes encoding all the other enzymes necessary to break down those particular sugars are turned off to save energy. These events include the enzymes necessary for the catabolic breakdown of lactose into energy. Now, there are three enzymes that are normally made by a cell when it comes to digesting lactose. And it makes sense that all these enzymes are made in the same ratio and controlled by the same genetic switch. So the control of multiple genes under the direction of a single genetic switch is known as an operon. Students need not be confused by the term operon. An operon is simply a bunch of genes, more than one, that is under the control of a single regulatory region of DNA, like a single promoter. Incidentally, operons are very common on the chromosomes of prokaryotes. So the Lac Operon, the one that we're dealing with in this video, is the classic example used by most texts. The tryptophan operon is another example, and the reason we discussed that in another video is that because it has a slightly different architecture. But there are many more, as we said earlier. But it's important to note that eukaryotes like us, we do not have any operons on our DNA. There were some discoveries recently that suggest that yeast cells have a few operons dispersed throughout their DNA, but that's not a common architecture. So let's spend a few moments looking at the arrangement of the information in the Lac Operon before we move on to look at its function. The Lac Operon, as we said earlier, consists of three genes placed head to tail along the same segment of the bacterial chromosome. So the first gene is called the LAC-Z gene, and that encodes a messenger RNA into a protein called beta-galactosidase. So the word "ase" on the end tells you that this is an enzyme, and we'll look at the function of this enzyme in a second. But immediately adjacent to the LAC-Z gene, without any spacing, is the Y gene, and the Y gene encodes another enzyme called permease. Permease allows lactose to enter the bacterial cytoplasm from the environment by placing into the membrane suitable facilitated protein transporters. And lastly, the third gene in the sequence is called the LAC-A gene, which produces a transacetylase, an enzyme whose function is still being deduced. This slide here shows us the actual activity of the beta-galactosidase. We should know that lactose is a milk sugar, and it consists of two rings, one of lactose and one of glucose, bonded together with a glycosidic bond. So what galactose does, it has a binding site on its surface for lactose, and it hydrolyzes the glycosidic bond, freeing galactose from glucose and releasing them into the environment. The glucose can then be broken down immediately to energy, and the galactose can be converted to glucose and then converted to that energy. Thus, when lactose is not present in the immediate environment of the bacteria, and it doesn't appear inside the cytoplasm, then it makes logical sense not to produce these three proteins. There's nothing for them to act on if there's no lactose inside the cytoplasm. Therefore, in order to save energy, these three genes are regulated and turned off when lactose levels are low, as we'll see in the next few slides. We just spent some time looking at the arrangement of the actual genes in the Lac Operon. What we haven't looked at is the switch, the genetic switch, which regulates the expression of these three genes. Let me take an aside here and talk about this word. So poly means many. Cistronic is another word for gene in genetics. So what this says is that this messenger RNA, this single piece of messenger RNA, carries the necessary information for producing three proteins from one messenger RNA molecule. Now, we haven't seen this before in this textbook or this course, but this is quite a common arrangement in bacteria when it comes to making proteins. So polycystonic messenger RNA is a single piece of RNA generated from a single segment of DNA, which can then encode multiple proteins. So technically, each one of these regions here must have an AUG site, as well as the Shine-Dalgrano sequence to which the ribosome will bind. And in the same vein, at the end of each colored region, there must be a terminator sequence which tells the ribosome to stop making polypeptide. So there will be three of those. Let's direct our attention to the all-important genetic switch. So this region here, also known as the regulatory DNA sequence, consists in this particular operon of three elements, two of which are shown here. So the third one is missing, and we'll talk about that later in this video sequence. So the first light green colored area is called the promoter, and we know what the promoter does. It's the region to which the RNA polymerase must bind and be orientated in the right direction to make this messenger RNA. But between the promoter and the first gene, the LAC-Z gene, there's a small segment of DNA, in this case known as the operator. And that has a very important function in determining if these genes will be transcribed into this polycistronic RNA. So these two elements, they sit side by side in this case. Now, just to be careful, sometimes the operator, this region here, the O, is found sitting inside the promoter sequence. But just for the sake of clarity, we'll consider that the two are side by side. That begs the question, what is the function of the operator region of the DNA? Well, this sequence of DNA is compatible with the binding of a repressor protein produced by another gene some distance away called the LAC-I gene. So the LAC-I gene, LAC stands for lactose, produces a protein, and that protein has a very important function when it comes to regulating the Lac Operon. The name of that protein is called the lactose repressor, and we'll talk about that in the next slide. But it's important to know that the lactose repressor has a shape that is compatible with the nucleotide sequence at the operator region of the chromosome. So the repressor has the ability to bind to the operator on the DNA. Before returning to look at the operator and its functionality, let's quickly expand on what we learned about the LAC-I gene. So the LAC-I gene produces the repressor protein, and this gene also has its own promoter, just here, that you can't see at this time. That promoter has no operator. The promoter is always on. It always binds RNA polymerase, and there's always a constitutive expression of the repressor in all cells at all times. So this repressor is never turned off under normal circumstances if this gene and its promoter are normal. A full understanding of the Lac Operon now hinges on students understanding what this repressor is. So the repressor protein has two binding sites. As we said earlier, one of the binding sites on the repressor is for the operator sequence on the DNA. But there's another binding site on the protein. This protein repressor has a second binding site, And that binding site is for the sugar lactose, the subject of the Lac Operon. Because this protein is able to diffuse through the cell, it's made by a ribosome and then it's released, it can diffuse and bind to any operator that it finds. And there's not just one molecule of this protein in the cell, there may be tens to hundreds, and each one of them is free to move around. So this is a trans factor, and it moves around and it bumps into both its ligands. It can bump into the DNA or it can bump into any lactose that's present. The binding of lactose causes an allosteric change in the surface of this repressor protein. So when lactose binds to one side, it causes a change in the configuration of the amino acids on the other side, and that change knocks off and changes the fingers that are used to bind to the operator. So when lactose is bound to one side of the repressor, it can no longer bind to the operator. So this is an important, very important concept. The repressor protein can bind to two different things. If it binds to lactose, which it prefers, then it cannot bind to the operator. But when there is no lactose in the cell, then the repressor maintains its original shape and it binds to the operator. So one or the other, not both. We are now in a position to see how the Lac Operon is regulated from the perspective of the bacteria. The level 1 label will become obvious later when we move into level 2 and level 3. Assume that you are the bacteria in the gut of a human being, and there is no lactose sugar in the digestive tract of that person. So what scenario can you imagine would exist at the surface of the Lac Operon? Well, this slide illustrates the arrangement of components that are present at that time. The RNA polymerase will be bound to the promoter sequence. The repressor protein will be bound to the operator sequence, and the polymerase will be blocked by the repressor protein from producing any transcripts. So let's go a little slower again, because this is key to understanding how the Lac Operon is regulated. Because the promoter sequence of the DNA has no mutations, it has the right sequence to bind the RNA polymerase. So RNA polymerase will bump into the sequence and stick around long enough in order to start to move to the right. However, at the same time, any repressor protein in the cytoplasm will be in a configuration where it has a liking for the operator sequence. So those molecules will bump into the operator, and through non-covalent bonding, the repressor protein will be found on the surface of the operator. Because the polymerase has to travel through the operator in order to transcribe the 3 structural genes for the Lac Operon, the block presented by the repressor is powerful enough to prevent and knock off the RNA polymerase. So if RNA polymerase cannot get through to the structural genes, there will be no polycistronic messenger RNA made from the Lac Operon. It is highly recommended that students pause the video and understand this very, very important concept. Once you've done that, then we'll move on to the next slide. So this is the situation that exists inside the bacteria when lactose is present in the environment and seeps into the bacterial cytoplasm. So lactose is represented here as the light blue molecule. So what happens? Well, the repressor proteins are bound by the lactose. So the lactose is the ligand for the repressor proteins. This binding of the lactose to the repressor protein results in an allosteric change in the shape of the repressor. That change is so powerful that it now no longer can bind to the operator. And if it was bound to the operator, it was now pulled off. The inability of the repressor protein to bind to the operator now frees the operator from any blockage. The RNA polymerase will continue to bind to the promoter, but this time it will have no hesitation in moving across this region of DNA and transcribing into the structural genes. So our polycistronic messenger RNA would now be made, and then the ribosomes would attach and then encode the 3 different types of enzymes necessary for digesting lactose. So the beta-galactosidase is now free to start attacking any lactose that's present in the cytoplasm and digesting it. The permease is now free to allow more and more lactose to enter the cell. This configuration will ensue until the levels of lactose begin to diminish. Once the lactose diminishes inside the cell, it can no longer stay bound to the repressor protein. The lactose will be pulled off and digested, and the repressor protein will take up its original shape and then be able to go back and bind to the operator and block any further transcription of the operon, therefore saving energy because there's no more lactose to digest. This is a very elegant and simple switch that nature has evolved that works wonderfully. Pause the video if you need to understand this further, because that understanding is paramount in moving forward to the next level of understanding. So once you understand the first cog, it's time now to introduce the second cog. The second cog involves the presence or absence of glucose. So now we're going to look at the combination of these 2 sugars within the cytoplasm of a bacteria. Here is some background information that's very important to understanding some of the complicated stuff that we don't have to know beforehand. So glucose, as we mentioned earlier, is the preferred source of sugar for energy purposes. So keep these facts in mind. So this is a very oversimplification of a very complex biochemical scenario, but good for us, it is. So when glucose levels in the cytoplasm are high, the levels of another small molecule known as cyclic AMP, very similar to ATP, are low. So this relationship exists. So when glucose levels are high, cyclic AMP levels are low. So we can use the analogy of a seesaw to understand that. So when glucose levels are high, the levels of cyclic AMP are low. And the converse is also true. So when glucose levels in a cell are low, then the levels of cyclic AMP will be high. So this relationship is very important for you to keep in mind. So what's the purpose of the cyclic AMP? Well, the cyclic AMP is a ligand for a protein called CAP factor. So CAP stands for catabolic activator protein. It's a general protein that's released when glucose levels are low inside the cell. And the CAP protein then has the ability to turn on alternative sugar metabolism pathways. And that would include the Lac Operon itself. So the CAP protein is an activator protein. So if you remember from the previous video, activator proteins increase the rate at which the RNA polymerase is able to do its job in binding to the promoter and transcribing genes. CAP needs a molecule of cyclic AMP to have the right shape and configuration to bind to its own DNA sequence called the CAP binding site. So the CAP binding sites are present on multiple genes, including the Lac Operon, and when they bind the CAP protein complex, they then increase the rate of transcription. However, if there are low levels of cyclic AMP in the cell, when glucose levels are high, then CAP protein will not be functional and it falls off the DNA, therefore minimizing the activity of RNA polymerase. The information in the previous few slides is summarized in this slide here. So in panel A, we reiterate the fact that when glucose levels are high, the level of cyclic AMP is low. And the reverse, when glucose levels are low, the levels of cyclic AMP are high. And the reason is indicated here in the activity of converting ATP to cyclic AMP. The second half of the slide shows us the relationship between the cyclic AMP and the CAP protein. So the cyclic AMP and the CAP bind together to produce the finished protein, which is able to then bind to the CAP binding site on or close to the promoter. In this case, it's shown sitting on the promoter, but in subsequent slides, we'll separate it and put it to the left as an assistant that helps RNA polymerase bind to the DNA. So finally, we arrive at this very key slide that was taken from a figure in the textbook. So it is a compound figure, and what we've done is we've broken it up into small segments to digest, and then we'll come back to it to relay the information it's presenting. So if we dissect that figure into relevant parts, the first thing to consider is the promoter region only. So the P region, the promoter, has one purpose: to bind the RNA polymerase. So if it doesn't bind the RNA polymerase, as indicated in these three panels here, 1, 2, 3, then obviously the rate of transcription is low, and the operon can be considered to be not functioning and is turned off. However, if RNA polymerase is sitting on the promoter, then there's a possibility that the operon will be on. Therefore, the RNA polymerase can perform some type of transcription. So that's what the promoter does. It binds to the RNA polymerase under certain circumstances. If we now isolate the next part of the regulatory region of the Lac Operon, we end up with the operator. So looking at the operator region only, it has, again, just one function. It can either be free of the repressor, or it can have the repressor protein bound to it. If the operator region of the Lac Operon is occupied by the repressor, then there is very low probability that RNA polymerase will be able to pass from the promoter, which is on the left-hand side, across to the genes on the right-hand side. So the repressor will block the passage of the RNA polymerase. That's a very important concept. What you see down on the bottom panel is that there is no repressor sitting on the operator. And in this particular scenario, RNA polymerase has the ability to bind to its promoter and move across and transcribe the genes. That is another very important concept. And then finally, let's look at the relationship between the cap binding site, the site at which the cap protein binds, when cap is associated with its ligand, cyclic AMP. So this scenario at the bottom here shows us that in certain circumstances, the cap protein can bind to the cap binding site, but in other circumstances, there is no binding of the cap to the cap binding site because the level of cyclic AMP is low. So summarizing, the top part here shows us what happens when the cell is feeding on glucose. There is no need to feed on lactose, so the cap protein is no longer sitting on the cap binding site, and the rate of transcription, the helping of the RNA polymerase to bind to the promoter, which will be here, is reduced to a minimum level. However, when glucose levels are low, then the cell has to turn to alternative sugars for its energy needs, and here the cap protein will be found bound to the cap site. Here the cap protein assists in bringing in and increasing the rate of transcription of the Lac Operon. So in conclusion, we have 4 possibilities for any particular cell when it comes to the Lac Operon. And here we have the totality of all the regulatory elements, the 3 of them, and how they work in unison to control the expression of these 3 genes, the Z gene, the Y gene, and the A gene. So let's take each scenario individually. Let's take the top row. So here, glucose is available to the cell, and so is lactose. And based on the logic that we understood at the beginning of this video, when glucose is available, then the level of lactose transcription should be low, because you want to dedicate all the energy to feeding on the glucose. So there is no cap assembled on the cap binding site. Remember the relationship: when glucose is high, cyclic AMP is low, and when cyclic AMP is low, cap protein can't do its job. So there's no assistance going on from the blue area. However, you would expect that the promoter site here would be bound with RNA polymerase. It is. So why is it not being shown in this figure? Because the rate at which the RNA polymerase binds to the promoter by itself without any assistance from the blue region is very, very low. So you're looking at maybe a couple of percent above being off. So in reality, the transcription of the operon is so low that it's considered to be baseline off. So that's a very important concept that's not really conveyed by this figure. Let's move to the second row. Here it says glucose is available to the cell and lactose is not. So again, looking at the cap site, the glucose levels are high, therefore the cap site will be unavailable because cyclic AMP levels are low, just like the case above. The only difference is because lactose is not present in the cell, the repressor binds to the operator. The repressor binds to the operator and guarantees more or less that if an RNA polymerase was to accidentally sit down on the promoter, there's no way that it can get past the repressor. And in this case, the operon is completely off. It's not even off at the few percent that the previous row was on. This is complete. The reason it's a fraction above zero is sometimes the repressor falls off accidentally and the polymerase can sneak past. The combination of sugars presented in the third row is that glucose is absent and so is lactose. So neither sugar is available for the bacteria to obtain energy from. So going back and dealing with each section independently, we see that for the blue area, we're interested in glucose. So if glucose is missing from the cell, cyclic AMP levels must be high. If cyclic AMP levels are high, then the cyclic AMP will be bound to the cap. The cap is now able to bind and assist the cap binding site in recruiting RNA polymerase. But at the same time, lactose is absent. When lactose is absent from the cell, it cannot pull the repressor off the operator. So here the repressors are attached to the operator. So both these proteins are sitting on the control element of the Lac Operon. And even if RNA polymerase was called in by the cap, the repressor would block any progression of the RNA polymerase into the coding part of the operon. So the operon is off. Finally, we have the last scenario where glucose is absent from the cell and lactose is present. So logically, the use of the sugar lactose should be encouraged. So let's see if that's the case. So dealing again with each section individually and independently, let's look at the cap binding site. So when glucose levels are low, cyclic AMP levels are high, cap protein is able to bind to the cap binding site, and we find it sitting right there. Also, when lactose is present in the cell, the lactose will bind to the repressor and pull the repressor off the operator. So there should be no repressor sitting here. And indeed, the repressor is missing from this section of the DNA. This means that the activity of cap encourages RNA polymerases, lots of them, to come and bind to the promoter, and it gives them a little push into the transcription of the genes. So as soon as one messenger RNA has been pushed along into the Z gene, another messenger RNA is recruited by cap, and then it follows behind. So that increases the transcription of the Lac Operon to its highest levels, and that is exactly what is needed by the cell in order to digest the lactose. So students are once again advised to stop the video here and to relate to all the information presented so far and explain to some colleague how this works. Once you're familiar with that, then we can move on to the final part. And the final part is level 3, where you can have multiple pieces of DNA within the same bacterial cell, and each piece of DNA may be carrying either part or the entire configuration of a Lac Operon. Having more than one copy of a gene is akin to being diploid, as humans are. So some bacteria, for some genes, can be diploid. So you'll see that terminology used in subsequent slides. So what other chromosomes can exist inside a bacterial that carry genes? Well, sometimes you get a second chromosome or part of a second chromosome accidentally being inherited by a bacteria or picked up from the environment through transformation. But most commonly, you find plasmids. Plasmids are small circles of DNA that carry genes that are very common in species of bacteria. And these extra pieces of DNA can carry the Lac Operon, as we mentioned a few minutes ago. So you could have two operons inside the same cell. And then the question becomes, using your knowledge and some knowledge that we'll present to you next, are you able to predict with some certainty the combination of genes that will be produced from those multiple Lac Operons? Let's begin with a simple analysis. So here we have a table of 4 different test tubes, each one with a different combination of chromosomes with different situations. In order to understand this table, we have to understand what the symbols mean. So the O stands for the operator, the Z stands for the beta-galactosidase, and the Y stands for the permease. Permease, the enzyme that allows lactose to enter the cell. As you can see, each of these locations on the DNA has a superscript, and the superscript is translated up here. So OC means that the operator for that piece of the chromosome is completely normal. So it has a normal sequence of DNA that allows it to work as a normal operator. The Z gene also has a plus sign in example strain 1. Therefore, that produces a completely functional beta-galactosidase. And the Y also has a plus symbol next to it. That means it too produces a completely functional normal permease. Now in the second example, in strain 2, we appear to have 2 chromosomes, and we do. So the /F prime stands for a plasmid. So these genes here after the slash are on the plasmid, and the first 3 genes are on the bacterial chromosome. So here we have a diploid condition for the Lac Operon. So that's for strain number 2. Strain 3 only has the bacterial chromosome, and strain 4 has the diploid configuration once again. So let's focus on strain 3. Strain 3 has an O with a C next to it, and the C stands for constitutive. That means that operator, and the C can only be applied to the operator, that sequence is altered so it can no longer bind a normal repressor protein. So let's look at the next 2 columns in the information provided. So this pertains to the enzyme beta-galactosidase. And what it means by non-induced is that if there is no lactose, what would happen to the production of this gene? And a minus sign means that the gene is not produced. What it means by induced is that there's a chemical added that mimics the presence of lactose, and what happens in that case. So whenever you see a minus sign, that means the beta-galactosidase is not produced under those conditions. And the plus symbol is used to indicate the presence of a fully functional beta-galactosidase under those conditions. Likewise, looking at the next 2 columns, we now refer to the Y gene, the permease. Once again, a negative sign means that the permease is not produced, and a plus sign means that the permease, a fully functional permease, is produced. So under non-induced conditions, is the permease produced, and under induced conditions, is the permease produced. Once again, non-induced means you mimic the circumstances where lactose is absent from the cell, and in the induced condition, you supply a chemical which mimics the presence of lactose. So I think we're now ready to tackle strain 1. So strain 1 could be a test tube that contains this strain of bacteria, which contains within it just the bacterial chromosome. And on the bacterial chromosome, the operator is normal, the Z gene is normal, and the Y gene is normal. So what do we expect to see if these elements are working as we learnt previously? Well, in the absence of a supply of lactose, we should not get any Z gene produced. Similarly, we should not get any Y gene produced. And what do we find? We find that under the normal circumstances, the operator prevents the RNA polymerase from producing any Z gene. Same thing with the Y gene. So that makes perfect sense. When lactose is mimicked inside the cell, we expect the repressor protein to be pulled off the operator, therefore allowing the promoter to encode these two genes. So we expect, in the presence of the inducer, both beta-galactosidase and permease to be produced. And that is exactly what the case is. So that is the wild type condition for a properly functional, inducible, that's controllable Lac Operon. Now let's tackle another test tube in which the bacteria contain not just the bacterial chromosome, the same as the one above, but they also contain a plasmid which contains a slightly different chromosome. Now on this chromosome, the operator is normal, the Z gene is defective, so beta-galactosidase cannot be produced from that segment of DNA under any conditions, but the permease is perfectly fine. So what do we see happening here? Well, as we know, the repressor can bind to this operator and that operator, because the repressor molecules are more than one, and they are trans molecules, trans factors. They can diffuse through the cell and bind to both these operators, blocking the passage of RNA polymerase. So when beta-galactosidase is non-induced, we expect that to mimic conditions where there's no lactose in the cell, and we would not expect to get any beta-galactosidase. Likewise, the same thing happens here for permease. So far, so good. Now, do we see any difference? Well, this fragment of DNA, the plasmid, will not produce any beta-galactosidase because the gene is broken. However, this gene on the chromosome is more than enough to produce enough beta-galactosidase to give us a positive result. So even though one gene is broken on the plasmid, the gene on the chromosome compensates for that lack of functionality. So we can say that the plus copy of the gene on the chromosome is dominant to the broken copy of the gene on the plasmid. So the Z plus is dominant to the Z minus. There's no difference in the Y gene. Both Y genes are fully functional, and if they are transcribed, they will produce both permeases from both those genes. Now we're in a position to tackle the third experiment, strain 3. So in this test tube, the bacteria contain just the bacterial chromosome, and on that bacterial chromosome, the Z gene and the Y gene are perfectly fine, but the operator is defective. Its sequence has changed so much that it can no longer bind the repressor. So the operator is said to be constitutive, as we mentioned earlier. Therefore, no repressor will bind there. So if no repressor binds, it doesn't matter if lactose is missing or if lactose is present. The genes are always available to be turned on by RNA polymerase, which binds to the promoter, not shown here, to the left of the operator. So you cannot block these genes under any circumstances. So what you expect to find is that beta-galactosidase and permease will be produced all the time. And this is exactly what is seen during this experiment. And then finally, we have another test tube, we call that strain 4, that contains this bacteria. And here the bacteria contains its chromosome plus a plasmid. What the students are required to do is come to this conclusion here. So let's look at how this is arrived at. If we look at the plasmid first, we notice that it contains a constitutive operator. That means that can never be turned off. So 100% of the time in these bacteria, these two products will be made. However, you can see that the Z gene, the beta-galactosidase, will be made full time and it will be perfectly normal. So we have two plus symbols. Whether you put lactose in there or you don't, it doesn't matter. The gene will be produced 100% of the time, all the time. However, the Y gene happens to be broken, and therefore it will never produce any functional permease under those conditions. So even though the operator cannot block the polymerase, it can only produce the Z gene 100% of the time, but not the Y gene. But we turn our attention now to the operator on the chromosome that operator is normal. It will bind the repressor when lactose is missing from the cell. But the product of its Z gene, which is sitting next door to the normal operator, that Z gene is broken. So it will never produce any functional beta-galactosidase. And the Y gene, the permease gene, is fully functional. So this operator, under certain conditions, will only produce Y, but will never produce Z. Because the operators in both cases impact only the genes next to them and not the genes on the other piece of DNA, i.e. the operator that's normal on the chromosome only has an effect on the two genes next to it. It doesn't have an effect on the Z and Y gene that are on the plasmid. So here, this particular operator, the DNA element, is said to be cis-acting. That means it can only influence its own genes and not the genes somewhere else. So this is a very important concept that is used to illustrate cis and trans in genetics. Here is a complementary diagram showing a fifth example of a situation. So we can pretend that the top section is the bacterial chromosome and the bottom section here could be the plasmid. In this particular scenario, the I gene is producing the repressor from both the genes. So we should have twice as much repressor in the cell, but that makes no difference. The amount of repressor is completely normal. Now, on the chromosome, everything is normal, and on the plasmid, we have a mutation in the operator. So let's look at the chromosome first. Everything should work as normal. So when lactose is not present inside the cell, the repressor will be found bound to the operator. So this repressor here will bind to the operator. Other copies of the repressor will bind to the other operator if they could, but unfortunately the sequence of the DNA on the second operator has mutated so much, changed so much, that the repressor cannot bind to that any longer. So that operator is not covered by a repressor, just the one on top on the bacterial chromosome. Because it's covered full time, the RNA polymerase is unable to get past the repressor on the operator. Therefore, the two genes on the bacterial chromosome cannot be transcribed. However, the RNA polymerases can bind to the promoter on the plasmid, and because there's nothing blocking their passage across into their two genes, and those two genes happen to be normal, they will produce beta-galactosidase and permease full time, regardless of the presence or absence of the lactose or lactose-like chemicals. This slide progresses the student into another scenario. Here we're not looking at the operator anymore. We assume that the operator is fine. We're now looking at the I gene and the I gene. So remember the Lac I gene makes the repressor. So the Lac I gene also can have a normal configuration. The plus means it's working completely normal, and the protein that it makes has both binding sites, one for the operator and one for the lactose. However, it can also produce a non-functional repressor, a broken protein, where it has no binding site for either the DNA or the lactose, because the protein is maybe truncated, it's defective. So under those conditions, we can now go through these four scenarios, and we should be able to explain what happens in the case of the repression of the Lac Operon. So looking at strain 1, in this tube, the bacteria contain a normal Lac repressor gene, and they contain a normal beta-galactosidase gene, and also a normal permease gene. And we can assume that the promoter is normal and the operator is normal ahead of these two genes. So what happens here? Well, when lactose is present, it will bind to the product of the I gene. And because it's normal, that will bind to the lactose and pull the lactose off the operator and allow the polymerase to transcribe the two genes. So the Z gene and the Y gene will be produced. What happens when lactose is absent? When lactose is absent, we can mimic that situation. Then the repressor that's produced normally by the I gene will bind to the operator and block the passage of RNA polymerase into these two genes. Therefore, when it comes to the beta-galactosidase, we should not see that, and nor should we see the permease. So the genetic label for that is that everything is working normally, and the product of the Lac I gene is inducible when the inducer is there, and is repressed when the inducer is absent. So it works completely normally. Let's turn our attention to strain 2. In this bacteria, the chromosome contains a broken Lac I gene, so it doesn't produce any repressor whatsoever. The promoter, the operator, the Z and the Y genes are perfectly normal. What happens in those cells? Well, as we know, the repressor is necessary to bind to the operator to prevent the RNA polymerase in certain circumstances from transcribing those genes. But there is no repressor in that cell whatsoever. It's missing completely. Therefore, the operator will never be blocked, and RNA polymerase has a free rein in getting through to these genes, and they'll be produced 100% of the time. So whether lactose is absent or lactose is present makes no difference, because the system cannot detect the presence of lactose or the absence of lactose. So we say that the Lac I gene constitutive, right? That means it's always in one configuration. It's always unable to turn the operator off, therefore the genes are always on. When genes are always on, as in housekeeping genes that we discussed in a previous lecture, then we can use the word constitutive gene expression. The next two experiments are some of my favorite because they illustrate something powerful. Let's look at strain 3 in this case. It's a diploid situation where the bacterial chromosome has a broken beta-galactosidase gene, but the plasmid has a non-functional Lac I repressor gene. What happens in this case? Well, we know that the normal gene produces protein that works perfectly normal. It can bind to both the operators on the chromosome and on the plasmid. Even though the plasmid itself is broken, the product of the normal I gene on the chromosome is able to transmit itself across the cell through the trans factor property and bind to both operators. So in this case, the learning objective is that the normal gene is dominant to the broken gene when it comes to the repressor. The normal repressor takes over from one gene the lack of ability encoded by the other gene, because enough is produced from the normal gene to compensate. In the final example given here by strain 4, we find a similar situation, but in this case the chromosomal version of the Lac I gene is broken, but the plasmid version is perfectly normal. In addition, the permease produced from the plasmid is broken, whereas the beta-galactosidase produced from the chromosome, bacterial chromosome, is perfectly fine. So this once again says that don't worry about the broken version of the I gene. It is compensated for by the fully functional version carried by the plasmid. So the plasmid version of the Lac repressor is produced in enough quantity to take over and cover both pieces of DNA. So we can say that the normal I plus is a transacting complement, a transacting factor. And I think on the next slide we can see that illustrated nicely here. So in this fifth example, we show that. So the bacterial chromosome has a broken repressor gene. It cannot produce any protein whatsoever, whereas the plasmid version produces a repressor, and that repressor is produced in enough quantity to bind to the two normal operators and control the operon in a normal manner. Finally, we have the very last type of mutation to consider. We've already considered that the Lac repressor can be normal, or the Lac repressor gene can be completely broken, but there are other mutations that are weird in the way that they work. So there is an I mutation that's been discovered that is called an IS. The S stands for super repressor. What that means is that the repressor cannot bind the lactose, the inducer. The mutation is in that part of the protein that binds the lactose. But the mutation does not affect the DNA binding side of the repressor. So the repressor can still bind to the operator even though it cannot bind somewhere else in the same protein to the lactose. So what effect do you think that would have? Well, the simple answer is once the repressor is produced, it will bind to the operator and it will never let go. It will never let go. And that's proven by these three strains down here. But before you discuss them, let's look at a graphical representation of the same thing. So here we have the IS, the super repressor. So this gene has a mutation of a completely different type that produces this repressor. And if you look very carefully, this end of the repressor has a different shape than the product of the normal I gene, which is indicated down here. So instead of having a square cog into which the lactose would fit, it now has an altered shape. So now the repressor, the lactose, cannot bind together. But the other side of the repressor is able to bind to the operator quite well. So this molecule will bind to this operator, and it will also bind to this operator and will never let go. So even though this repressor is perfectly normal, this one is more powerful and it will bind to this operator also. Therefore, this mutation is said to be dominant. It takes over regardless of what the other gene is trying to do. So the product of this gene is dominant and it suppresses the entire two sets of chromosomes, the bacterial chromosome and the plasmid chromosome, preventing any transcription from taking place whatsoever. So strain 1 is perfectly normal and it works normally as we decided previously. It's strain 2 that gives us an insight into what the super repressor does. So here the repressor is made and it binds to the operator and it never lets go, regardless of whatever conditions pertain. Therefore, the beta-galactosidase and the permease will never be made. The system is always repressed. And in this final example, let's look at a partial diploid. So in this particular test tube, we have a bacteria that contains a chromosome that produces the super repressor, and it contains a plasmid that produces a normal repressor. And both the genes are absolutely normal, the beta-galactosidase and the permease. And what do you expect to happen? Well, if you revisit this figure on the next slide, this is exactly what you're seeing over here. So in this case, there is no way that you can induce the production of either one of these two genes. Now that you have been exposed to the deep understanding of the Lac Operon, it's time to test your knowledge. I recommend that you stop the video here before answering this question and practice your skills. Once you have the answer to this slide calculated, you may look at the next slide. The next slide contains the answer. Pause the video to reflect. Here's your next question. Please pause the video now so you can answer this. Here's the answer to the previous question. Here's your next question, which is slightly harder. Please pause the video now to work on this problem. Here's the answer to the problem. Now that represents the end of this module. Thank you so much. Bye-bye.